So we know a little bit about their life history and their physiology, but we still don't really understand anything about how they generate these terrestrial locomotor behaviors. So we explored several aspects of how they do this because we want to understand how these fish are actually accomplishing the feat of terrestrial locomotion. So the first two questions, how does this fish transition from water to land? And once it's on land, how are they capable of locomoting there? Are mostly exploratory studies. We then have some hypothesis-driven experiments looking at the forces they generate when moving on land, followed by the motor pattern that they use to drive these behaviors via their axial muscles when they're out of the water. So the first thing we have to do is collect our specimens. So we go into the mangrove swamps. I am completely covered. It was 103 degrees that day, 90-something% humidity, and you don't want to get mosquito bitten. So my hands were eventually covered up, but it's hot and crazy, and that's what you look like, but it's worth it because you get fish. So we go to our little Kmart and get our animals. So on the top left, so Miami is right up here for reference. These are the Florida Keys. So here is Key Largo. We go to a slew of places along the Keys. And then my favorite place in the world, here is Lighthouse Reef Atoll. It's about 50 miles off the coast of Belize City in the Yucatan. And to blow this up, this is about 20 miles the way the crow flies. And we sample in the mangrove keys, such as Long Key in the south, and Northern and Sandboard Key in the north. And if you kind of squint and use your imagination, I think Lighthouse Reef looks just like a Kmart. So that was very convenient. So these dots in red denote the locations where we collected all these fish from multiple populations, genetically and geographically distinct. So here is what our field site looks like on Long Key. The two red circles denote some of our sites. This is also the base of the operations for our Lighthouse Reef class. We stay right here and snorkel all these places throughout the atoll. So here is a picture of a drone that Max Messenger, a grad student with Miles Silman, was flying at about 1,000 feet over us, while myself, Dr. Miriam Ashley Ross, and a slew of students from a year ago were sampling in the mangroves. Let's take a dive into the canopy, under the canopy. This is what the red mangrove swamp looks like. Species Rhizophora mangle. So these fish are occupying these watery habitats, and they have to negotiate around all of these prop roots. And pockmarked throughout these prop roots are a bunch of crab burrows. So on the left, here's the opening to a crab burrow occupied by a land crab, Cardosoma guanhumi. It's about the diameter of a softball. Now on the bottom right are the cup traps we use, and you can see them outlined in these red circles of how we capture the fish. So no, I didn't steal this from the pit. That's the cafeteria at Wake Forest, if you're unfamiliar. We actually ordered these. They weren't repurposed. We take a little funnel, insert it into the cup, little 16 ounces, and we drill holes in this, and then we stick this into the crab burrow or in between a bunch of prop roots, wait for 1 to 2 hours, and allow the fish to hopefully swim in, not swim out, and we'll get anywhere from 1 to 6 fish at any given time. You'll notice there's no water. The tide has gone out. And this is the optimal time to capture these particular fish. So I'll just pass this around if you want to see. All right, so you have to be very careful where you place your prop root because saltwater crocodiles occupy a lot of these areas. Miriam and I luckily still have our hands, and we're not the prey consumed by this little guy. We wait an hour and a half, and we get to explore wonderful places such as this while we are waiting for fish to occupy these cup traps. We then revisit the cup traps, and the first thing we do after we collect them, hopefully, is take a little fin clip from their tail fin, and we send that to a colleague of ours, and we genotype every single individual. So you can't actually see it, but I'm manipulating the fish, and a bunch of students, some of whom are in the audience, were assisting. Here you can see some of the cup traps for size. So we then put them in little whirl packs. It's basically a glorified Ziploc, and we line them up with about 30 mils of water and take them back to the States in a nice inconspicuous carry-on bag. So we're now back in the lab. Here is my colony at Wake Forest, and there is one fish occupying each 32-ounce container. They're in brackish water. It's temperature controlled and everything like that. They're all marked, and we then go to work. So to remind you what the first question was, we want to explore how these fish transition from water to land. So through a series of natural history papers, we know that these fish actually pursue prey items out of the water. So we were able to train these fish to capture this pinhead cricket on a modeling clay platform. So the fish, what you're going to see in the successive video, is moving from the left to the right. It's usually not this wavy. We have two high-speed video cameras, one in the dorsolateral position and one in the lateral position, which is where your eyes are currently in. So you're going to watch this fish launch out of the water from left to right, and you can see that he overshoots the clay platform and falls into the water on the other side. This behavior occurs at about 27 body lengths per second. Now, that's incredible because if you are familiar with salmon, they leap out of the water at about 10 body lengths per second. So these fish are performing at 2.5 times that rate, which is amazing. They have a takeoff angle of approximately 47 degrees. So to have maximum horizontal displacement, you would predict 45 degrees is what you would achieve, and that's exactly what we see with these fish on average. They also reach a height of about 3 to 4 body lengths. So from the aerial position, you'll see how this fish actually generates this particular behavior. So in the still image sequence, starting on the top left, they make an S-bend with their body. So they bring their tail up toward the cranial region, and they accelerate out of the water. And this all occurs in about 50 milliseconds, and that is the launch behavior. This occurs about 55% of the time of their three major behaviors. So the second most common behavior is the pounce, which is basically a truncated launch. So you'll notice that there was a pinhead cricket at the water-land interface. It made purchase with the item, immediately takes it into the water, violently thrashes it around to force it down its buccal cavity for eventual consumption. And this behavior only occurs when the animal is feeding. So the third most common behavior is the squiggle. This is published. It is now a scientific term. Use it as much as you want. It's basically like an army crawl. So you can see that the pectoral fins, the side fins, are making purchase with the substrate. They will use that to pivot around with their body as their fulcrum as they then continue to move forward from water onto land. And you can see there is a lot of swinging going on with their tail, where about 20% below the head is staying somewhat still through that entire process. So here are some data. There's a lot to walk you through, so let's go. So here we have this nice little silhouette. This black symbol denotes the head, followed by the red triangle, all the way down to this magenta X representing the tail. Now the axes are the absolute space in which the body parts are moving forward. So let's start with the head region. So this open symbol represents the first position of movement. You can see that there's lots of lateral undulation with the head. You can see right here 20% of the way through the body, which is close to its center of mass. You can see very little lateral movement. It's staying mostly still as the head swings side to side. Starting with the tail over here, you can see it makes this figure 8 type movement. And that's reminiscent of both aquatic and terrestrial fishes, such as eels and ropefish. It's a pretty common behavior. And we see that with the caimar. So basically the take-home is lots of movement with the head and the tail, and the mid-body is staying mostly still as it squiggles forward. So you see this starting here, moving toward the top of the slide. That's because everything is moving forward. Now here we're measuring amplitude. So this 0 axis corresponds to this horizontal orientation of the body. The symbols represent the same thing as the previous slide. So when the fish's body moves toward the left, that we just denote is a positive. And when the fish's body moves toward the right, we have that as a negative. So on the bottom, you can see through an entire cycle, wave cycle of the squiggle, there's a lot of undulation, again, with the head in black, and the tail, you can see, also has a lot of undulation. And at 20%, you can see, again, it's staying very, very consistent along this horizontal 0 axis. So not much movement with the mid-body, lots of undulation with the head and the tail as it squiggles forward. So lots